Peace be upon you, grade 8 students. In this video, we are going to discuss these objectives. First of all, dear students, let us make some revision. What is the role of natural barriers? Yes, natural barriers prevent the entrance of pathogens into the body. What is the largest organ of the natural barriers? Excellent, the skin is the largest organ of the natural barriers. When can a pathogen cross the skin? A pathogen can cross the skin in case of a wound or a burn. In both cases, there will be an opened area in the skin, and through this opened area, pathogens can enter into the body and cause infection. Remember very well, infection is the invasion and multiplication of the pathogen inside the body. So, after a burn or a wound in the skin, which is the largest natural barrier, microbes become able to cross the opened area in the skin and might cause infection. Other than skin wound or burn that represents a good situation for the entrance of pathogens, some pathogens can cross other natural barriers to cause an illness. What is the solution for this illness? Yes, it is by taking medicines. What is the role of these medicines? These medicines help to destruct the microbe after infection. So, some illnesses require the use of certain medications to prevent the entry of the microbe or to destruct it after infection. But how can we ensure prevention of infection or destruction of microbes after infection? Which means, how can we help ourselves avoid infection or destruct the microbe after it enters the body and causes infection? This is what we are going to discuss in this video. In some cases, the immune system does not interfere to defend and protect itself against microbes. But there are several aids or helps that do this job. You have to read the title of each paragraph in your book, page 34-35, to pick up the names of the 4 sorts of aids of the immune system. The sorts of aids of the immune system represent several kinds of helps that help the body defend itself against microbe. The 4 sorts of aids of the immune system are antisepsis, asepsis, chemotherapy, and antibiotherapy. The role of these sorts of aids of the immune system is to prevent infection or destruct microbes after infection. Let us start with explanation of antisepsis. Please read the following paragraph and observe figure A below. The skin is the most important barrier against infection. In case of a wound, it is necessary to wash abundantly the wound with water and soap, then to apply a solution called antiseptic that slows down or prevents the multiplication of microbes. The most common antiseptics that are used externally are eosin, hydrogen peroxide, alcohol, and iodine. They are toxic for bacteria but not for human cells. After cleansing, it is important to cover the wound with a sterile dressing. In this figure, it is important to apply a solution called antiseptic on a wound. This antiseptic is against wound infection. Referring to the paragraph, what solution is applied on a wound? Yes, this solution is antiseptic. What is its role? The antiseptic slows down or prevents the multiplication of microbes. List the 4 examples of this solution. These examples are eosin, hydrogen peroxide, alcohol, and iodine. As a summary, we can say that in a case of a wound or a burn, apply antiseptic solution on the affected area. The antiseptic solutions prevent and avoid the multiplication of microbes. The 4 kinds of antiseptic solutions are iodine, alcohol, hydrogen peroxide, and eosin. What happens if the operating room and the surgical instruments are contaminated with microbes. Yes, these microbes will be transferred to the patient and cause infections in him. So how can we ensure the sterilization or the destruction of microbes of the operating room and the surgical instruments to prevent the infection of the patient with microbes? Dear students, keep in mind that the operating room and the surgical instruments should be completely without microbes to protect the patients from getting infections. Let us discover how we can destruct the microbes in the operating room and on the surgical instruments. Please read the following paragraph and observe figure below. This paragraph is about asepsis. Asepsis is the total absence of microbes. It is mandatory, for example, during surgeries. It is provided by the sterilization of the operating area and the surgical instruments. Sterilization is the destruction of microbes. It can be obtained by several means, including treatment of instruments at very high temperature and the use of disinfectants such as bleach for the floor. Answer these questions referring to this paragraph. What is asepsis? Asepsis is the total absence of microbes. How is asepsis provided? Asepsis is provided by the sterilization of the operating area and the surgical instruments. Define sterilization. Sterilization is the destruction of microbes. List 2 ways of sterilization. Sterilization is provided by treatment of instruments at very high temperature and the use of disinfectants such as bleach for the floor. So asepsis, which is the total absence of microbes, is needed in the operating room and for the surgical instruments. Asepsis is provided by sterilization, which is the destruction of microbes. Microbes can be destructed either by treatment of instruments at very high temperature or the use of disinfectants such as bleach. As a summary, asepsis is the total absence of microbes, which means to make a medium completely without microbes. Asepsis is required for the operating room and the surgical instruments to avoid infection. But how to ensure asepsis and make the medium without microbes? Asepsis is ensured by sterilization, which is destruction of microbes. There are 2 main ways for sterilization. The first way is the treatment of instruments at very high temperature. It involves putting the surgical instruments in a dry heater sterilizer or oven, for example. The high temperature kills all the microbes that might be present on the surgical instruments. The second way is the use of disinfectants, such as bleaches for the floor and operating room bed. The disinfectants kill all the microbes that might be present on the floor or on the operating room bed. In both ways, we can ensure the total destruction of microbes. We have studied about 2 sorts of aids of the immune system, which are antisepsis and asepsis. Let us move to chemotherapy and antibiotherapy. Some diseases require the intake of medicines to be treated. What do we call the treatment that involves the use of these medicines? Let us read the following paragraph and observe figure C below to discover the name of these treatments. Chemotherapy and antibiotherapy. Chemotherapy is the treatment of the infected patient with chemical substances that are toxic to microbial diseases. So chemotherapy is when the patient is given specific medicines that are directed against a certain microbe. In particular, antibiotherapy is the use of antibacterial medicines called antibiotics. Antibiotics are medicines that are directed only against bacteria and not against any other type of microbes such as viruses. Figure C shows some medicines. These medications are precious aids to the immune system. Refer to the paragraph and define chemotherapy. Chemotherapy is a treatment of the infected patient with chemical substances that are toxic to microbial diseases. And define antibiotherapy. Antibiotherapy is the use of antibacterial medicines called antibiotics, so the medicines that are directed only against bacteria are called antibiotics. Pay attention, dear students, that chemotherapy and antibiotherapy take place after infection, which means after the entrance of the microbe into the body in order to treat the disease. But asepsis and antisepsis prevent and avoid infection. Asepsis and antisepsis are measurements done in order to avoid the infection. As a summary, we can say that certain illnesses must be treated by medications or drugs that destruct microbes after infection. These treatments are chemotherapy and antibiotherapy. Both treatments involve the use of medicines, but in antibiotherapy, these medicines are called antibiotics because they are directed only against bacteria. The antibiotic leads to the destruction of the cell wall of the bacteria, so antibiotics are only directed against bacteria. And once the cell wall of the bacterium is destructed, the bacterium dies. Penicillin is the first discovered antibiotic, and it is used against bacterial infections such as respiratory tract infections, mouth infections, and throat infections. But how was penicillin discovered? A scientist named Alexander Fleming discovered the first antibiotic penicillin. A green yeast, which is a kind of fungus called Penicillium, accidentally invaded a bacterial culture studied by Fleming. Fleming noticed that the bacteria has died in the area around Penicillium. This is the bacterial culture studied by Fleming. Notice the huge number of bacteria in this bacterial culture and specifically in this area. And this is the Penicillium colony, which is a green yeast that contaminated this bacterial culture. Notice the disappearance of bacteria in the area around the Penicillium. So in this area, the bacterial growth was inhibited or prevented. Why do you think the reason behind the death of the bacteria around the Penicillium? Can Penicillium kill these bacteria? Of course, yes, because Penicillium produces a chemical substance called penicillin that is able to kill these bacteria. 10 years later, penicillin was extracted from Penicillium and used as an antibiotic to treat some bacterial infections. Let us read this paragraph about antibiogram. Penicillin is the first antibiotic. It was discovered by Alexander Fleming when one of his bacteria cultures got accidentally contaminated with a green yeast called Penicillium. Fleming then noticed that the bacteria has died in the area around the yeast. 10 years later, a chemical substance called penicillin was extracted from Penicillium. Nowadays, there are a lot of antibiotics, but how can we determine the best antibiotic against a certain type of bacteria? Let us discover by reading the following paragraph. Nowadays, there are many natural and synthetic antibiotics. It is often necessary for the treatment of a patient to know what antibiotic best kills the bacteria he or she infected with. So to treat a patient against a bacterial infection, he should be given an antibiotic that best kills the bacteria he or she infected with in order to treat him correctly. For that purpose, an antibiogram is performed. Then, why is an antibiogram performed? An antibiogram is performed in order to know what antibiotic best kills the bacteria the patient infected with. But what is an antibiogram? An antibiogram is a culture of bacteria isolated or taken from the patient in the presence of different antibiotics. Here there is a jellified medium containing the bacteria isolated from the patient, in addition to 4 discs. In each disc there is a specific type of antibiotic. So there are 1 type of bacteria and 4 types of antibiotics. We need to know which type of antibiotic best kills this type of bacteria. What do these dark areas around each antibiotic represent? These dark areas around each antibiotic represent the areas which are devoid or without bacteria, which means that in these dark areas the bacteria has died due to the antibiotic. Among the 4 antibiotics, deduce the most efficient antibiotic against this type of bacteria, knowing that the most efficient antibiotic is the one that makes the jellified medium around it more devoid of or without bacteria than the other antibiotics in the disc. Excellent! Antibiotic 4 is the most efficient antibiotic against this type of bacteria because it makes the medium around it more devoid of bacteria. So what is the least efficient antibiotic against this type of bacteria? Excellent, it is antibiotic 1 because it makes the medium around it less devoid of bacteria. Dear students, pause the video and solve this evaluation. Write the appropriate name of the sort of aid of the immune system next to each sentence. Treatment of antibacterial diseases by medicines called antibiotics. This sort of aid is called antibiotherapy. To make a medium devoid of microbes or the total absence of microbes, it is asepsis. Treatment of microbial diseases by chemical substances, it is chemotherapy. Use of an antiseptic to a skin lesion to avoid multiplication of microbes, it is antisepsis. Also pause the video and solve this evaluation in which you have to classify the 4 sorts of aids of the immune system into the 2 following groups: preventive methods or curative methods. Antisepsis and asepsis are preventive methods because they prevent and avoid infection, while chemotherapy and antibiotherapy are curative methods because they destruct the microbe after infection to cure the disease. At the end, dear students, thank you for your attention.